talk will be slightly different from what we have been hearing in the morning, and I will try in the next 20, 30 minutes to convince you that some of these tools, AI tools, could probably be useful for some of the data technologies that you are developing here. So basically, what I will be discussing is about how biases coming from different data modalities, from medical data, can be introduced and can fuel the development of AI models. And in particular, just to start with the very quick motivation about this type of work, of course, we are all aware that the great technological advancements nowadays in terms of technologies are transforming medicine. Now we have access to an atlas of data per patient across different scales. So we can have pathology images, we can have radiology images, we can go at a cell level, even at a 3D with multiplex immunofluorescence images, spatial transcriptomics, spatial proteomics. So we have a huge atlas of data around patients that, if exploited correctly, can give us quite a lot of information in terms of trying to understand the disease, understand biomarkers for the disease, trying to see if a person can respond to specific therapies, try to tailor therapies to specific people, and achieve what we call personalization in terms of medicine. So all these great technologies give us promise that we can advance in terms of discovery, and we can advance also in terms of diagnostic and prognostic tools that can be used in clinical practice for clinicians in their decision-making process. And on the top of that, of course, we have AI, where we believe it can play a key role as a tool for exploiting the wealth of this data, which, of course, it's definitely not an easy task, and that's why we haven't seen the huge success stories of AI for health applications, because of the fact that, of course, the medical domain, the health domain, is quite complicated. Apart from the fact that all these technologies, if you change across sites, across hospitals, you will have data sets coming with different biases, different artifacts. At the same time, we are usually working with small clinical cohorts, in particular in rare diseases. So we are not in a setup of cats and dogs where we have a lot of images and we try to infer from this amount of images, but we have very limited data that are coming from patients, that are coming from rare diseases, that are coming from challenging setups for AI. And on the top of that, of course, I don't need to motivate you that much, but as we know, biology and human health in general, it's a quite complex system where you have dependencies across time, across scales, starting at the gene level, at the cell level, in terms of a tumor, at the microenvironment level, and overall at the systemic level. So you have all these complicated dependencies that are quite complicated to take into account with classical AI tools. So if I just do a very quick one-slide summary of where we are in terms of AI nowadays, we have seen all these quite successful AI architectures that have been developed across the years. But if we want to abstract a bit, the main intuition behind all these architectures has been that their design has been guided by explicit biases coming from the application domain. So if we think, for example, of perceptrons, if we go quite a few years back, the main assumption there was the regularity of the function. CNNs, you have images, you want to capture the fact that you have translation invariance in terms of the image. So if you have one cat appearing in one part of the image and another one appearing in a different part of the image, you would like to understand that we are talking about the same feature. RNNs, time component, it's another inductive bias that has led to the design of this sort of architectures. Graph neural networks are architectures for network type of data where basically the prior there has been that of permutation invariance, meaning that if you change the position of a node in a network, that shouldn't change how you represent that node. So basically, in all these setups, we have seen a lot of success in terms of machine learning, in terms of AI models that follow these inductive biases. Of course, if we think of medicine, we are talking about genes, we are talking about cells, we are talking about many different processes, as mentioned before, that are quite much more complex than what we could model with current technologies. In particular, if you think of spatial proteomics data, for example, you move from RGB 3-channel images into images with quite a lot more than 30 markers. each of the markers expressing a different protein. So how can you extract that information? How can you define technologies or architectures that can capture that information? So the way that we like to approach this sort of problems, of course, from an AI perspective, is that we would like to think of developing AI tools in which we introduce the inductive bias coming from the application domain. Meaning, if we have prior knowledge about the data, if there is knowledge coming from biology, from clinicians, that knowledge should be introduced, or physics, that knowledge should be introduced in the design of the algorithm in order to be able to develop more generalizable and robust models that can eventually be used either for, as I said, biomedical sort of discoveries, identifying new biomarkers, or as diagnostic tools, supportive tools for clinicians in their decision-making process. So, and what could be, just to make it a bit more concrete, what could be these inductive biases? So in this talk, I will mainly focus on one specific aspect that is about networks as a natural inductive bias coming from different application domains. If we think of quite a few biomedical data, and in particular imaging, there is an underlying network complex structure that can represent the data. I'm looking forward to the talk of Dimitri later. Maybe he will say a few things about neuroscience there, but the brain typically, it's the structural connectivity of the brain, the functional connectivity of the brain. These are things that many people are actively working on. In terms of spatial biology, pathology images, the cell-cell interaction, the spatial interaction of the cells is something that we know that has a prognostic value in terms of response to different treatments, how cells interact with each other. And then if we go into images like for example, angiography, we have a clear geometry there coming from the fact that you have an artery tree. So all this geometry can be considered as a mesh or a network of different arteries that are connected to each other. And then of course, are the non-imaging applications, but for the sake of time, I will not spend a lot of time on those. Molecules, for example, protein-protein interaction networks. You have these complex dependencies between entities that, if exploited, could give you information in terms of extracting useful information from this data. And of course, we have these networks, and graphs are just a mathematical tool for modeling these networks, for modeling these complex relationships. So I will be focusing mainly on two clinical use cases or big disease areas in which we are very interested in. And the first one is cardiovascular disease, and the second one is cancer. Of course, I don't think I need to motivate them more. I can, as we know that more than almost 50% of deaths nowadays are coming from these two diseases. So cardiovascular diseases are the first cause of death, and cancer, of course, is the second cause of death. So if AI can achieve some success in these two big areas, of course, that would be something extremely useful for humanity. So to make it a bit more precise, so what I will be talking today is two use cases, if you want, or two problems coming from collaborations that we have with clinicians, and see how these problems fuel the design of these AI methods, particularly graph-based AI methods. The first use case would be in the domain of cardiovascular disease, and in terms of predicting future cardiac events from a collaboration that we have with the cardiology department of CIBM. And the second one will be more on PET-CT imaging, modeling metastatic lesions, and modeling predicting response to the therapy. And that's another large collaboration that we have with the University Hospital of Geneva, CIBM, the University Hospital of Zurich, and the HSSO and the Swiss Data Science Center. So let's start with the cardiovascular disease part. So the first application, it's about predicting future cardiovascular disease. So whenever someone is going with some symptoms, basically with what they will do in clinics, they will have coronary angiography. It's an invasive procedure in which you get this sort of modalities, this sort of X-ray images, in which you can see the geometry of the anatomy. And typically, I'm oversimplifying things, but typically what a clinician will decide in practice is we'll see if there is a narrowing in terms of the stenosis, we'll quantify that narrowing, and we'll decide if that narrowing should be treated or not. So the diameter of that narrowing or the diameter of that stenosis, it's a clinical indicator that is used in practice for cardiovascular diseases. What we envision to do is to try to use this information coming from angiography together with information that we might have from prior knowledge coming from hemodynamics, from physical knowledge, in order to be able to develop AI models that will combine expertise coming from clinician data and mathematical modeling. And as I said, that is an ongoing collaboration that has been there for quite a few years with CIBM, with the cardiology department of CIBM. So as I said earlier, if we go into clinical setups, there are two clinical matrices that are quite important. First is the diameter stenosis that I mentioned before, the narrowing in terms of the artery. And the second one is the flow, the fractional flow reserve, the FFR, that is basically an indicator of the flow, how it drops in terms of the artery. So these are the two things that are quantified in practice. So now what we are proposing is really to take into account this intuition of the stenosis, of modeling the stenosis and the narrowing of the stenosis in an AI framework that will be able to take an angiography and be able to predict what is the risk score of the patient of having a heart attack or myocardial infarction in the next few months or years. So basically what we have done is that from these images, we construct, segment these images, we annotate the part that is the lesion, the problematic part, and we map it into a graph that will capture explicitly the geometry of these lesions. There we develop some AI tools that are based on graph neural networks, so generalization of neural networks into networks and graphs. And we use these tools in order to get, as I said, the predictive score. Then we fuse it with additional information coming also from flow, from FFR, etc., clinical information. So I will not go into much detail into the modeling, but just to give you an overview of how it looks like for those who are more interested into the technical aspect. So we start with images, we construct a graph that will capture the narrowing of the arteries. We design a lesion level prediction that is based on graph neural networks. We introduce clinical features there that are related to the age, gender, et cetera, and then we have a score at a lesion level. We can then move from a lesion level design to a design that is more a patient level, in which you would combine these different lesions with a mechanism that is updated with some attention sort of mechanisms that would adapt to different numbers of lesions per patient, and be able to model the risk of a patient developing a heart attack in the next couple of months. So if we just briefly check the results of how it works, the results are quite encouraging. We have been testing it in two clinical cohorts, FAME2 and another smaller cohort from CIBM. Basically, I don't have the time to go into all the details, but if you check the curves down for both data sets, the ROC curves that are a metric of evaluating the performance, the higher the better, and you will see that by taking into account explicitly this geometry, with the purple line you get a performance that is much better than having a purely image-based model or having models that are based on clinical evaluations based on the FFR or the diameter stenosis. And then going one step further, so far we've been talking about modeling the narrowing with the graph, but basically what we can do is to move from these 2D images, from the angiography, from these X-ray images, into a 3D reconstruction of the arteries. And this is something that is done by our colleagues at the Center for Imaging at EPFL, where basically they take two views of this angiography, they reconstruct what we call a digital twin, the 3D model of the arteries, in which then we can run hemodynamic simulations and have a representation that is more patient-specific, enriched with knowledge from hemodynamic simulations. So in that way, we unify anatomy with hemodynamics in order to be able to have a richer representation per patient. So we can construct 3D digital twins in which we can run CFDs. Of course, CFDs are quite complex. In order to obtain, they take a lot of time. Basically, one way that we're proposing to overcome the limitation of having to explicitly to compute these thermodynamic simulations is to use our prior knowledge from physics. So we know that the blood fluid dynamics can be modeled with a Navier-Stokes equation. And we have also laws. I will not go into the details. You can ignore the maths there. You can just trust me. They are taken from the literature. So there are equations modeling the flow dynamics and modeling also the pressure drop in terms of the arteries that can give us an approximation of how we expect this drop to be. So basically what we propose is a two-step approach that will couple modeling of physics with patient data and the digital twins that we mentioned before in order to have a first step where you would basically learn hemodynamics from the data by not using labels in a self-supervised way, as we call that, but by using only this notion of physical equations that we know for data. So the prior we introduced there is the notion of geometry coupled together with physical equations that will help us train an ML model that would predict features that are aligned with the hemodynamic constraints that we know from the physics. Once we do that, and we can do that for a lot of data because you don't need labels for that, we can then take our small clinical cohorts, fine-tune them with labels, with the future event predictions, for example, and get a performance that hopefully is much better. And that's the overall idea of the framework. And if we go a bit more into the details of that, basically, in order to summarize what we said, we take an image, we compute a digital twin of this image. Then we have another step where from these digital twins, we can generate synthetic digital twins by basically combining digital twins of different patients in a meaningful way for this specific application. And then we can create an artificial large data set in which we can do this sort of physics-informed self-supervised learning in order to pre-train with the physical losses that I mentioned before. So we can pre-train with quite a lot of synthetic data generated from patient data. And then once we have this pre-trained pipeline, we can fine-tune it using, again, graph neural network-based architectures that I will not go into the details in order to achieve some predictive performance on downstream tasks that are related to future events. Just to show you a few curves of how it looked like. Again, it's the ROC curve. The higher, the better. And we can see that if you focus on figure A on the left, with the blue line is what we achieve with this digital twin representation together with physics introduced into that. And if we have done quite a lot of ablation study in order to understand how much physics is helping, how much digital twins are helping. Since we're coupling all this information together, I don't have much time to go into the details, but indeed, if we do all of these components independently are helping in terms of getting a higher performance for predicting future cardiovascular events, and in particular myocardial infarction. And as a matter of fact, I talked about virtual data generated from digital twins of patients. And basically, we can see that in terms of, these are different matrices, accuracy, F1 score, that is another matrix that we use in terms of basically classification, ROC curve, etc. So all these scores, the higher the better in all cases. And we can see that if we train a model just with clinical data, that would be the blue line there. If we train this model with some sort of pre-training, still on clinical data, with physics-informed constraints, you get a slightly bit better performance in an orange curve, and then with a green curve, if you introduce more virtual data. So by doing all these steps together, you get a performance that is quite important. And in terms of explainability of these methods, because we introduce these physics-based constraints, we learn features that basically capture what we would expect from the notion of hemodynamics. So in lesion areas, in areas that are problematic, we have a pressure drop that is on average much higher, than in lesion areas. which means that the features that we get are quite informative. And you can see it also how the pressure will change. In the red box are basically the lesions, and we see that there is a difference in the pressure along exactly the part of the lesion that we are trying to model. So in the next very few minutes, I will just present a second use case. I will go quickly through that is more into the cancer side. And basically, there we are working with PET-CT images. And the idea is to be able to improve the patient stratification and be able to predict if a patient is going to respond to a therapy or not by learning the tumor dynamics. So the overall idea is that we have a set of PET-CT scans from patients that have been treated with immunotherapy and would like to model how these lesions evolve over time, appear, disappear, split, etc., using AI. So the idea there is that basically the prior that we would like to introduce is that we want to treat these lesions together. Usually what is done nowadays is that we focus at the lesion level or we aggregate all lesions together, but without not taking into account the systemic behavior of the lesions as a system. So basically we're modeling the lesions as nodes of a graph, and we have a set of graphs from each time point from metastatic lesions over time. And we would like to capture these lesions, this system, in such a way that we're able to detect states such as hyperprogression, progression, pseudoprogression, etc. So be able to monitor better and predict if a patient is going to respond to the therapy or not. So I will not go too much into the details for the sake of time, but the type of framework that we model there are based on state space models enriched with notions coming from graph learning in order to be able to combine these longitudinal lesions of the data into a patient representation feature that would be later on represented or lead to a state representation, meaning if a patient or a lesion is progressing or not. So the goal is that you input these features, it could be radiomics, features characterizing the lesions, and be able to predict the next time point, meaning how these lesions will evolve, but also the state, the underlying state of how the disease is evolving over time. We have tested it on the melanoma cohort coming from the University Hospital of Zurich, and we're currently testing it also from cohorts coming from CIBM on patients with melanoma. And we seem to have quite interesting results in terms of detecting the regimes where indeed the state of the patient is changing, and in terms of predicting this change of points. So also here on the, I will not focus too much on the numbers, but if you just check the colors, at time 5, for example, you have a change in the regime, and you would see that gradually it is red in the beginning because the change is quite abrupt, but then it converges quite quickly into identifying the correct state of the patient, basically. So it's an ongoing work which we are trying to extend also to other type of diseases and other cancers. So to summarize, I hope I try to convince you that AI could be useful in some of these applications, and in particular graphs are a tool, a model that could be able to capture some of the complexities in the data. Of course, a graph is just a prior that you introduce in the data, and by no means it means that every biomedical problem should be casted as a graph. But in use cases where it makes sense to capture these dependencies between different entities, then definitely it could be something worth considering. And I would like to finish by saying that I think in all these type of applications that I mentioned before, collaborations are really key in order to be able to identify the use cases where it could be meaningful to apply AI and to be able to develop AI tools in terms of identifying biomarkers, in terms of developing prognostic diagnostic tools, but at the same time, trying to understand how we can build the right tools or the right cohorts, how can we acquire data in such a way that they can be useful for AI. And towards these two aspects, we are trying to put quite a lot of effort. and try to build a lot of collaborations. One of those is what Jonas mentioned before, that is a National AI Initiative for Precision Oncology. The goal, it's led by the EPFL AI Center in collaboration with ETH Zurich, with 4 university hospitals, cantonal hospitals, private clinics. Then we have pharma companies involved, Roche, smaller startup, etc. But the overall idea is to build a platform for precision oncology where AI can be useful in different stages of the disease. It can be useful from identifying biomarkers to defining which treatment is the best for each patient in such a way that the profile of one patient can fuel or can improve the treatment of the next patient. And all that in a secure environment that we will try to set up over the next 4 years. In the aspect of how to bridge, how to make clinics for AI, if you want, or how to be able to interact in order to build data sets, cohorts in which AI can indeed shine in collaboration with the cardiology department of CHUV and also HG here in Geneva. Basically, we are launching the Swiss Cardia study that is a cohort, a longitudinal cohort in which we'll be able to monitor patients for 4 years and capture all different aspects of the disease, from CT imaging, MRI, but also to lifestyle data, genomics, food related initiatives, etc., in such a way that we have a cohort where indeed AI could bring some meaningful information. So, let me finish by acknowledging all my collaborators. I presented quite a few different aspects of them. So most of the work that I present, the technical part, was conducted by Jeremy, my PhD student, and Shao, who was a postdoc and left us for a faculty position in China. And then, of course, all the collaborations from CHUV, SSSO, the University Hospital of Zurich. And thanks also to all the funding who has supported this work. So.